Yes, sir.
Good. So in yesterday's class, good morning everyone. Let's start this. And in yesterday's class, we discussed a few things, and we were trying to build a motivation or to see the need or the significance of stem cell biology. So in today's class, we will review what we have already discussed, or at least the summary of it, and then we will discuss a little more about the course, and then we will go into basics or fundamentals of developmental biology or embryology. So as is the case with every organism, humans also want to live long, to exist long. That is the priority. But then there are also other things like we need to be healthy as long as we live, to reproduce and execute our free will or our instincts, to be able to be happy, right? And when we do that, we have a lot of problems. And in that, we have come to a situation where we would be having problems. It's likely because of other species or because of other humans. There could be problems with during birth or reproductive defects, immunity problems, malnourishment, pollution, accidents, and aesthetics. These are some of the ways that you can be affected. These are all physical problems. There are also other problems which are psychological in nature, depression and other things. So we need to take care of these, and at least as a focus for, or the importance of stem cell biology comes in all of these cases where our physical existence has become a trouble, and how to rectify that is based on or basing on regenerative medicine. And then in that center, for regenerative medicine, the center or the basis comes stem cell biology. We also discussed in this case that in every problems, the solution should, at least in the physical attributes of our organisms, there is form and function, right? So we need to take care of these two things, and out of these two, function is more important. Then comes the form, right? Say, for example, if you are going to an expensive restaurant, five-star hotel, for example, then you would be, you, the chef is making you a dish, right? Then there are two things about what a chef does. How delicious is it? First question, first point. And the second is how healthy, delicious and healthy. And the second part of the chef is to plate plate it in an attractive way, right? So if you see some of the food shows, they will show you how they decorate the food in such a way that it is appealing to the eye, right? Taste has two things: visual as well as the tongue. In this case, plating is kind of form. How do you present it to somebody who is observing it? And the function of the food to be delicious and healthy is to make good quality food and tasty to the tongue. Even if somebody is closing their eyes and tasting it, they should still be able to feel good about it, right? That is how you can think of. It's like, what would you choose is something like this. There is a gift, you have a present, which is packed very well. My drawing is bad. And there is another parcel which is not packed properly at all. Which one is somebody going to choose? They will choose for the attractive packaging. And but the point is, what is the content of it, right? At the end, whatever is being chosen, the content of it will matter, right? The function is the content of it. How you wrap it, how cleanly you do it, how attractive is the form. Why I'm talking, as we discussed earlier, if you want to replace an organ, then one thing is it should look like an organ that is normal in nature, but it should also function. And in that case, we were discussing about some of these things where here the function, the progress is towards function, but form-wise, it's not so appealing. We inherently have social stigma to everybody who has something abnormal. I mean, little bit of deviation from what the norm is, then we find it weird. So that is where everybody is supposed to have some things, and their organs or other things are supposed to look a certain way. And when they don't, then the society reacts differently. So form is important as well. Function is for that person, the person's ability to do stuff. Form is for the onlookers to feel normal among the people. Both are, of course, going to end up to the person itself. So the idea is, if we want to, you might have already learned, you might have had a course on prosthetics, or you may be having, that is trying to put artificial limbs and other things, robotic arms and so on. Those are fantastic inventions or the paths. But what is ideal would be to regenerate the limbs and put them on. Yesterday or today I was watching, I read a news where a person who has received a limb, a hand of another person, a donor, who I think has died or so. I haven't read all the scientific details of it. And that person went to meet the kin or the family of the person who has donated the hand. And it's nice progress that has been done, right? That is donating the limbs. But how often can somebody donate limbs? I mean, one has to wait. We will see in this course also several of these donations that are possible. And the goal of regenerative medicine is kind of regenerate. That means to be able to produce it outside and put it onto the person, or to somehow stimulate the regeneration of the arm on a body. Say, for example, diabetes. We have problems with several things. It is hyperglycemia, that is increased blood glucose in the body. That could be caused because of many things, including diet. We have bad diet. It is highly rich in glycemic index causing food, which means that we are having a lot more carbohydrate than what we should have. Carbohydrates, like say many of the things that we had. What are we born as humans? If you take, we were the earliest humans, for example. What would have been their diet? There was no agriculture, right? So they would be eating. I mean, if you want to draw what would have been their diet, we should look at our closest evolutionary ancestors. They would be chimps and gorilla. They would be eating a lot of fruit. That is, we are frugivores. Fruits is one thing. We also eat roots, something like, and tubers and other things. They are mostly carbohydrates, but they are actually coming along with many other things. And then they would have been eating meat by hunting. Chimps do hunt as well, and they eat other monkeys and other things that they can catch. So this is the diet of typical humans. They may also have eaten some leaves. Okay, what are we eating now is chips, uh, snacks like or biscuits, cookies, you can call them, cakes. How many of you are salivating now? Chocolates, so many other things. And if you talk about lunch and dinner, it's mostly rice. And we do take vegetables and other things, but they're a very minor proportion, right? So this is the modern day type of diet, which is not good for our health. Meaning the sense it is good, but too much or every day of it is not good. It just shoots up the blood glucose in the person, and eventually the body becomes trained or gets used to the amount of insulin, and it stops absorbing the glucose, and that is how you have more blood glucose in the body, glucose in the blood. I hope you have noticed your grandparents and maybe some of your parents who may be diabetic. The sense that is, it has become a normal thing in all of our lives. Another important thing I would also want you to focus is no exercise. Say if we were the ancestral one, the ancestral human being, if you're born some thousands of years, then we would have been walking a lot. We would have been running a lot. We would have been fighting a lot. Okay? What are we doing now? We are sitting a lot. And is there any time we are actually stretching our whole stretching our body to the limits of the possibilities? Like, are you playing any sports actively every day, all through the day? No. We have about, in the, say, we take out 8 hours of sleep, and then we have some 16 hours. Say 2-3 hours or so we can put it for daily routine things. And the rest of the time, or you can say 4 hours, okay? All the rest of the time, if you look at, we are mostly sitting. They call in the somewhere I read this that they call sitting is the next smoking, something like that. To mean the meaning of it is sitting is equivalent to smoking. It's as bad as smoking, right? Smoking causes a lot of problems, and so does sitting for prolonged time. So that is one of the bad bad things we have. I mean, sitting as such is not a problem. The implied meaning is that inactivity is the problem. Inactivity is the problem. Right now, if you are sitting and sleeping, I wouldn't mind if you're going around walking while you're listening if it's on your mobile. Uh, the ideal way to spend a day is something like this. Not that I'm following absolutely, but I'm trying. Okay, that's what we can all do. So if this is the duration of your day, and every hour you should have spent about 10 to 20 minutes standing, walking, or somehow moving your legs. The general notion that if we exercise here 45 minutes or something, or in the evening 45 minutes, that's good. Good enough is the notion, and all this time sitting. That is not right. It is still bad. You don't have to do 45 minutes of any sports or anything. You just, for every hour, if you are spending 10-20 minutes walking, standing, and all these other activities other than being in rest, you're going to have a very good metabolism. Okay, so consistent activity, consistent daily activity, that is what you should remember. um, consistent daily activity. What I meant, it is the activity should have been spread all throughout the day. And then you are in very good, you will be in good shape, your metabolism will be good. And then these lifestyle diseases, diabetes is one of the lifestyle diseases. There are other reasons too, but we will focus for the time being. And then, of course, the stress, that is mostly because of social pressures. That is, either you want others to view you in one way, or others want you to deliver something to them. Right? Most of this stress comes from the society, meaning that uh, either we are afraid, or we are conscious that somebody might be thinking low of us or bad about us, and we try to make it right and try to please them, or try to show a better image of ourselves, and we fall into the trap and then build stress in ourselves. The second one is somebody wants you to deliver, like I might give you an assignment and I'd say you have to submit the assignment. That is a pressure too. And or somebody might want you to come to a wedding or some function that you might not want to go. That is a pressure too. Everything will mount up and eventually will build a personality in us such a way that we don't know how to say no, and eventually that becomes a lot of stress. So you should know how to have fun, how to be upfront, how to be honest and say maybe not now, some other day, something like that. Okay, but what we are talking about is all these things are contributing to a lot of diseases which we refer to as lifestyle diseases, such as diabetes. And it is very prevalent. My parents had it, my grandparents had it all too, and I'm likely to have as well. And unless I keep up my metabolism, keep up my daily lifestyle, it's not going to be a good thing for any of us. So I hope why I'm stressing this is, if you are, as you are at home and you're listening to this class, if it is possible, any of my classes or anybody's classes, you can use your earphones, mobile, and walk around instead of sitting in one place or sleeping on the bed. Just saying. So what is the reason for type 2 diabetes? There is type 1, we will discuss those, but type 2 is one diabetes, it's hypertension because of inactivity or consistent inactivity. The metabolism goes down, the body, skeletal muscles and liver tissue stops responding to amount of insulin that is present. We will discuss these things in greater detail, so this is not the topic here. I just wanted to imply about the importance of stem cell biology. So we will only stick to those details. So if the problem is there is in the blood, you have high concentration of glucose, and you want to you if there is high blood glucose or hyperglycemia, it is bad for many tissues, including eyes, kidneys, brain, heart, and so on. So how to make it low? It is blood glucose increases because of um, sorry, insulin, lack of insulin. Insulin is the hormone that triggers or signals the cells, the target cells, like skeletal muscle cells and liver cells, to take up the glucose from blood, thereby bringing the blood glucose lower. Right? So the insulin, either the insulin is not produced in enough concentration, or the target cells are not responding to the amount of insulin. Right? If that is the case, I mean, both are dependent on insulin or insulin response to insulin. In the case of insulin, ideal, what we're trying, if you want to make an ideal remedy, say for example, if it were deficiency of insulin, okay, type 1, in that case, we should be able to produce insulin and give insulin to the person. How can we produce? typically through injections which were produced by recombinant DNA technology. You produce a lot of insulin into the vials, and then the person gets injected with it so that there is insulin deficiency can be controlled. But is it ideal? It's not ideal. Why is that? Because how much insulin should be given? When should it be given? How many times this person has to take injections, and how long should he do, he or she? Right? Instead, think of a remedy like this. You, the insulin loss is because of the beta cells of the pancreas are dead, so which produce the insulin, therefore you don't have. Say, for example, this is pancreas and there are no beta cells. What if we can make some beta cells and put them into pancreas, and they produce insulin as and when required? That would be the ideal remedy, isn't it? So we are in all of regenerative medicine or stem cell biology, the goal, actual or the ultimate goal, is to regenerate or make the body as it was functional before, right? If there is a defective part, we want to rebuild it. If there is a loss, we want to make one and put it there, or allow the cell, the body itself, to make a new part of its own. I hope you understood what we wanted to do. So in this course, stem cell biology, fundamentals of stem cell biology, we will read about embryonic stem cells in the first unit. Second unit is about pluripotent stem cells, epigenetics, reprogramming, and so on. There will be some techniques in that. And also techniques are there in all of these units, okay? Methods and how to do those stuff. And in the third unit is completely dedicated to cancer stem cells. We will read a great deal about it. So I don't think you would lose a lot from cancer biology. But of course, cancer biology has its own. When you read something in a specialized way, it is a different thing. But you will have good enough introduction to cancer biology as well. And then in the final one, we'll have the applications, where we will read about how it can be used and all. One thing I want to tell you upfront. I don't think there is any proven and completely certified stem cell based therapy for any of the diseases. Majority of it is in research or clinical trials. Okay, keep that in mind, and that is where it will help us um, be close to the reality of it. This is a therapy or strategy approach for the future, and that's why it's important to study more, so that we will be able to build. If it is already done, why should we read about it, isn't it? And that's why stem cell biology and technology becomes central to majority of most people's research and passion, actually. The prescribed textbook is little old, but it doesn't mean there is any deficiency. It's called Strengths in Stem Cell Biology and Technology. I have shared you the copy of it, I think, if material is already there. I want you to understand the content level. The content level is for professionals and practitioners. Okay, so it is good enough, good for, even if though it is at 10 years old, it is still good for us. But I have also shared other books which will be useful if you want to read more. There will be some more articles that you can always find and read. So in this course, we will generally talk about principles and what can be taken ahead in the future. There will be at points where we don't know complete details, but that is all right. Okay. So all of stem cell biology we have to start with always the basics of developmental biology, because we haven't studied anywhere as much. Maybe in +2 or 10th you may have studied a little bit. So I'll just review it so that we can recollect these. I want you to focus one thing. Uh, uh, do an exercise along with me during the course. Anytime you are listening to the course, I would want you to take a piece of paper and sit with it. And when you get a new term, please write it. Biology or any of the sciences in general is, or science is generally, you will have problem with jargon. That is so many technical words and so on. Or I would rather call it as glossary. You supposed to build a glossary. So when the class is happening, try to write only that word. At least write that word so that you will be able to memorize. If you can memorize the words, technical terms, then you know all biology. That's what it means actually. So please pay attention and try to do it in such a way so that you will find biology interesting and comfortable. Because in the future, when there is a sentence, if you are reading a sentence, and this book is for professional and practitioners, right? It's not a textbook. It is not a typical textbook like you start with questions, you start with, you start addressing or describing what is what, and then you'll have a question, something like that. It's not a textbook. It consists of reviews. Reviews means they have read a lot of research papers, combined them, and made it into and then try to synthesize some information, and the authors may have their own opinions in that, which may be true, which you may agree or disagree, which might have been correct at that point of time, and which may not be relevant at this time point. So we have to go with an approach of balance, and we should know where to go and not. Especially if it is a review, each sentence will have at two or three technical terms. And when you see these technical terms and you don't know the meaning of it, we lose the contact with the text or the subject itself. And then we will have a lot of problems with biology. Okay, so try to do this exercise. Take a paper and pen and write down any new technical term so that you'll feel comfortable in the later classes. You'll get used to it, and then you'll be able to write research papers better as well. Okay, so we will learn about the fundamentals of developmental biology. The first one is zygote. You all must have known about it. Zygote is the first cell, first diploid cell, 2N, formed through fertilization of an egg, and this is sperm is N, the egg cell has N haploid, and then it gives rise to a diploid cell. And that is the first diploid cell formed out of the gametes, and that is called as zygote. And it is totipotent. We will tell totipotency here means that the whole, if a cell can give rise to a whole organism, right? Like we all have come from one zygote. Any cell that can give rise to a whole organism, living organism, then you call it as toti. Toti, you can take it for total or full potential. You can total potential. You can think of. The next stage is morula. The zygote will divide into multiple cells, and it will form a mass of cells, something like a sphere of cells. Each of them looks similar. They are identical looking. That is morula. That's called as morula. And then we have blastula. That is where the first differentiation would have happened. What do you mean by differentiation? So I will try to write it here. Differentiation is you have a cell, and if it is giving rise to same type of cell, with all physical properties as well as biological properties, then they both are identical, right? These two cells are identical. If if the same cell gives rise to a, a different type of cell, okay, something like this. Now, cell A and cell B are not identical. Cell A has differentiated into cell type B. It has changed its properties, either physical or biological properties, and that is called as differentiation, right? Cell A has different types of functions and properties compared to that of cell B. Then you call that differentiation, changing from one state to another state. And in this case, from morula, the first differentiation happens in the sense that some of the cells will be will form something like a circle, like a football bladder, and you will find all these cells outside here that I've drawn here, they would have looked identical. And 1 second, and some cells inside might look different. Okay, so you have two different types of cells in blastula. You have the outside trophoblast cells, and the inside you have a cell mass that is called as inner cell mass, ICM, inner cell mass, that's given here. And trophoblast is the outside whole circle. Trophoectoderm, you can call it also. That is blastula. And I hope you understood what is differentiation. In cell biology, you must have read it. Then there is the formation of gastrula, where you have the formation of primordial germ layers, where you have the blastula, the inner cell mass will, I'm not drawing, okay, let's put it this way also. You have the trophoblast cells, right? All of them look similar. And the inner cell mass will give rise to 3 germ layers. Okay, there will be variations between different, the embryology or developmental biology of different organisms will have variations. Like it's different for frogs, it is different for humans, and so on. What I'm trying to do here is to give you some idea about some of the things that are common in most of the organisms. So during gastrulation, inner cell mass will give rise to 3 different layers. Okay, you call them as primordial germ layers: ectoderm, endoderm, mesoderm. Okay, meso will be in the middle, ecto and endo, something like that. That's the mesh, and then eventually that grows into the fetus. It will be something like this, and then organogenesis, the formation of organs, and then the infant is born. Infant will be, once the infant is born, birth, and then the growth will happen. And in adolescence, there will be sexual maturity, and then in adult, we have reproduction, and then sometimes, or often in humans, what will happen is we'll have a lot of parental care, resources to generate resources for progeny to survive and thrive. It's like this. Our parents work hard, they give birth to us, and they still keep earning because they want to provide for us. They still keep earning so that we don't have to, we will, they, so that we will be ensured of a long and happy life, because money makes many things. And eventually we will all become old, and then we need to be taken care of and hope that the our progeny actually takes care of us. And then death death could happen in natural consequences where the person just the body and becomes old and old, it becomes thin and then they die out because one or more of organ failures or in, inconsistent functions. Sometimes it could be because of accidents, and sometimes it will be because of diseases. Diseases, there is one quote somebody was saying, if we all live long enough, we will all die out of cancer. We don't have to agree with that, but that is how prevalent cancer has become in a way. I hope you understood some developmental biology, and I hope you got the some of the technical terms. You know what is fertilization, the fusion of male and female gametes. And you know zygote, that is the cell, first diploid cell formed upon fertilization. I've also described what is meant by totipotency. It is the formation, it is the ability, potential of a cell to give rise to a complete organism, complete viable organism. If you think it properly, only in humans or higher organisms, only the zygote is totipotent. No other cell can do that. If you take a plant, for example, in majority of the cases that is true that they can give only plants, the seeds can give rise to a whole plant, or the embryo, which again forms from the zygote, can give rise to a whole embryo. But you can take a stem of some things, and like in potato, for example, you can take a small piece of potato and put it into soil, it can give rise to a complete organism. You can't do that with humans. There are some organisms which can do a little bit like planaria and other things, but we will discuss those things at a later time. So you know about spermatogenesis and oogenesis. I will go with this and we will see. Yeah. So spermatogenesis and oogenesis. We discussed in the last course about the about mitosis and meiosis, about how chromosomes actually are segregated and so on. Here too we are going to discuss the same, but you have to be very thorough with that. It's not that this is very essential, but we have to discuss some things about X chromosome inactivation and many other things. For that, you have to know of these things. I'll describe these two and then I will stop. So there is parametogonial stem cells that is responsible for the production of the sperm. You're saying stem cells. We haven't described what is meant by stemness. We will do that in the next class. For the moment, try to know about spermatogenesis and oogenesis. And there is also primordial germ cell. You call them as PGC. At times we might have these abbreviations. In the initial classes, I would avoid them. I will try to speak in full, but as we go, these keeps accumulating, because of which we will be forced to use the abbreviations. So primordial germ cells. Do not confuse it with the primordial germ layers. Here, in this case, in gastrula stage, I said primordial germ layers, right? And you would also have primordial germ cells. They actually form at a later stage, but we'll just continue there. Primordial germ layers are ectoderm, endoderm, mesoderm. Primordial germ cells, okay, or I would put it here itself. So if there is a person and the person's whole body is made up of, say, you call that as somatoplasm, isn't it? All of the body is called as somatoplasm. And only the gonads, or the germ cells within the gonads, or the gamete forming cells, belong to the germplasm. So all of somatoplasm is made from the primordial germ layers, ectoderm, mesoderm, endoderm. All of the germplasm is made from primordial germ cells. Okay, keep that in mind, and that is how we will think of primordial germ layers are different from primordial germ cells. They both form during the early developmental stages, but then primordial germ layers give rise to the whole of our body, physical body, and the germplasm or the primordial germ cells gives rise to the gamete forming cells. So here we are talking about primordial germ cell in embryos. So they undergo mitotic divisions, they form spermatogonial stem cells, and they undergo mitosis, and they form spermatogonium and several other mitotic divisions, and then there is the first formation of primary spermatocyte. Primary spermatocyte will undergo meiosis. Until then, you see here and here, it's just mitosis. They are increasing their numbers, that's it. Then at 2n, we have, that is diploid primary spermatocyte, and upon undergoing meiosis 1 and meiosis 2, we have formation of early spermatid. And these spermatid undergo cell differentiation. As in the previous classes, I told you what is meant by differentiation. Say, if you look at the early spermatid and sperm, their physical properties and biological functions are completely different, right? The early spermatid cannot swim, whereas sperm can swim because it has a tail, right? So that is a simple example of differentiation, and that is how finally sperm forms with a head in which the whole of the genome is tightly packed around protamines, and there is a long tail to allow swimming from one place to the another place. Upon copulation, it has to move from the through the uterus to the fallopian tube to meet the egg and then fertilize. It's a long distance uh, to swim, so it needs a um, tail like this. So the oogonium, yeah, I will stop here because I don't think I should. But otherwise, I'll just try to implant some of the technical terms so that you will remember. Right, primordial germ cells, oogonium, mitosis will happen, and then you have the primary oocyte, which will start undergoing meiosis. There is something different in humans in where gametogenesis happens. In males, so the spermatogonial stem cells, they remain inactive until puberty, somewhere around until 15 years or 16 years. So there is no meiosis yet. No meiosis is happening. They are arrested in spermatogonial. They remain like this in G0 phase. And only upon maturation or puberty, the male will start producing sperm. Whereas in oogenesis or in the female, the eggs or the primary oocyte already starts undergoing meiosis 1, even when the baby is still in the womb itself. Okay, and then it is arrested. It enters into a cell cycle arrest until the individual becomes or undergoes puberty. And there are more details that we will discuss in the next class. And I want you to read the reproductive biology lessons from 10th or +2 if you haven't read. We should all be very, what you call that, um, we should know about our own bodies. In many schools, there will be some shyness to speak about this or learn. now it is essential that everybody should know. We are biology students. If we don't know our own biology, what do we know? That will be injustice to our own education itself. So take it professional, and I would want you to see the fundamentals of reproductive biology from +2 is enough, or you can watch YouTubes and try to know the biology of it. How do our bodies change? What is produced and what happens? Okay, I'll stop here. I think we have reached the time. We'll meet in the next class. Hello?
Yes,
Any
sir.
Yes,
questions?
sir.
You can also ask any questions if not. Okay, then we'll meet in the next class. Okay, take care and
Thank you.
have fun. Yes, bye-bye.
Thank you, sir.
Thank you,
Thank you,
sir.
sir.
Thank you, sir.
Yes, you had a question, somebody?